Good morning, students. Hope you all are fit and fine by God's grace. As we all know that due to lockdown, it is not possible for us to continue our regular classes from college. So the only option left with us is to study from home itself. So, I don't know whether you have covered this topic or not. If you have covered also, no problem. Just revise along with me. There is no harm if you revise once again. As more as you revise, the more easier the concept becomes to you. Isn't it? So, today we will study about physicochemical events in fertilization. So, what is fertilization? It is the process of union of male and female gametes, that is sperm of male and ovum of female. Both of them fuse to form a diploid cell that is called zygote. Okay, so what is the site of fertilization? As we all know, that fertilization occurs in fallopian tube. So the exact location where the fertilization take place is ampullary isthmic junction. Okay, so in most of the exam this option is not been provided. So if it is not given, then always tick the option ampulla. Never ever tick the option isthmus. Okay, most of you get confused with both these terms. So isthmus is the, you can see the fallopian tube in the picture. So the narrow region is known as isthmus, and it dilates. As it comes, it dilates a bit. This dilated portion is known as ampulla, and the last region that is attached to ovary, you can see the finger like projection. This portion is known as Fimbriae. Okay, so in the, you can see in the picture, in the ampulla the sperm has reached where the egg is present from before, that is being released from ovary. It is waiting there to for fusion, so that the sperm can come and fuse with it. So you can see at that place fertilization take place. Okay, so what is the time of fertilization? As we all know that normal menstrual cycle is of 28 days. Okay, and ovulation occurs in the middle of menstrual cycle, that is on the 14th day from the first day of last menstruation, when usually a secondary oocyte is released from either ovary alternately. Okay, so why do we call it a secondary oocyte? Why we don't call it a ovum? Because it has not undergone because we call it a secondary oocyte because it has not undergone the second meiotic division. Okay, that is, it has not undergone metaphase to anaphase 2. It has done up to meiosis 2 and it has stopped it after being fertilized with that sperm. It will continue its further division. Okay, so it is waiting for the male sperm, so that the male sperm can come and fuse with it, so that it can undergo more division, further division. So and remember, the secondary oocytes of female remain alive for 24 to 48 hours, and at that time, if it is not fertilized, it gets degenerated, whereas sperms that is deposited in vagina of female reproductive tract remain alive for 72 hours. Okay, then the next topic, pathway of sperm to fallopian tube. So after copulation, when the sperm is ejaculated, it first reaches the vagina, then it reaches the cervix, you can see in the picture, then through uterine cavity it reaches the fallopian tubule. Okay, you can see first vagina, cervix, uterine cavity, then fallopian tubule, and in the fallopian tubule it goes to ampulla where the egg is present from before, and there the fertilization take place. So before going to the process of fertilization, just we need to know the structure of secondary oocyte. Otherwise we will not be able to understand how the sperm means how the sperm penetrates and all. So we can see here the outermost layer of secondary oocyte. The granulosa cells are appearing as radiating rays to us, and this outermost layer is known as corona radiata. Okay, so the inner to this corona radiata is present another layer, and this layer is known as zona pellucida, and the innermost layer, that is the plasma membrane of oocyte. Okay, the plasma membrane of secondary oocyte also has a one name, and it is called vitelline membrane. And between the vitelline membrane and the zona pellucida, you can see in the picture that there is a one white space, and this white space is known as perivitelline space. Okay, this is known as perivitelline space. So just see pathway of sperm to reach the secondary oocyte. So first of all, the sperm will invade the corona radiata, then it will go to zona pellucida, then through perivitelline space it will reach the plasma membrane of secondary oocyte, and lastly it will go to the cytoplasm of secondary oocyte. Okay, just see in the picture, it is shown clearly. So the process of fertilization. So when the sperm is being ejaculated, we all know that millions of sperms are ejaculated. Okay, so the one which will travel faster will get the chance of fertilization. You can see in the picture, see millions of sperms are there. Okay, here there is no millions, but many sperms are there, but only one. You can see in the picture that only one has got the chance to fertilize. Okay, so after it reaches the secondary oocyte, I think most of you have gone through the structure of sperm. So after it reaches the secondary oocyte, the sperm has an acrosomal, means the head of the sperm has a acrosomal cap. Okay, acrosomal cap. So the acrosomal cap releases enzymes. Okay, the name of the enzymes are hyaluronidase and acrosin. They have specific function. Okay, and both of these enzymes are collectively known as sperm lysins, remember. And what does the enzyme hyaluronidase do? It causes the dispersion of corona radiata cells so that the sperms get access to zona pellucida. You can see there, see the first layer is corona radiata, isn't it? So what does this enzyme do? It causes dispersion of the corona radiata, the first layer, and the sperm enters the first layer. Then what it will get? It will get zona pellucida. Okay, so the second enzyme comes into action that time, that is acrosin. What does the acrosin do? The acrosin creates a hole in the zona pellucida so that the sperm can enter the zona pellucida and reach the perivitelline space, and finally it can go to the secondary oocyte. Okay, so what happened after the hole is being created in zona pellucida and the sperm enters, some chemical changes occurs at that time. Okay, and what happened, some chemical changes occur, and which causes the zona pellucida to thicken and get hardened, so that what happened, the rest of the sperms, that the millions sperms can't get access to it. Okay, so what means, mainly it prevents polyspermy. Okay, it prevents the entry, means the I said no, that zona pellucida gets thickened and hardened, and it forms fertilization membrane. Okay, so what is the function of this fertilization membrane? This fertilization membrane prevents the entry of more than one sperm. Okay, it prevents polyspermy. So what happened after fusion of this sperm has entered the zona pellucida, then it has finally reached the vitelline membrane. So after it fuses with the vitelline membrane, the only the head portion of the sperm enters the cytoplasm, that is enters the cytoplasm of secondary oocyte, and the tail is left behind. Okay, just remember, tail also don't go inside. Okay, tail is left behind, and only the head region that contains nucleus and the centriole gets inside. Okay, so immediately when the sperm enters, the secondary oocyte undergoes second meiotic division to produce ovum and second polar body. Okay, so the second polar body which is formed is very small in size and has very little cytoplasmic content. Okay, it has very little, so it gets degenerated. It has no function. It gets degenerated. And what happened, just remember, the sperm nucleus that enters, it has its own centriole and nucleus. Okay, but the sperm nucleus do not have a cytoplasm. Okay, and the female that is the secondary oocyte has this nucleus also and cytoplasm also, but the oocyte do not have its own centriole. Okay, just remember. So what happened, so after the second meiotic division, it forms ovum and second polar body. So ovum, what it do, ovum nucleus enlarges to form female pronucleus. At the same time, sperm nucleus also swells up to form male pronucleus. Okay, and the male and female both are haploid. Okay, so this both haploid pronuclei fuse together and completes the process of fertilization, forming a diploid nucleus which is known as zygote. Okay, both of the male and female haploid pronuclei fuse to complete the process of fertilization, forming a diploid nucleus called zygote. And the zygote that is formed has two sets of chromosome. Okay, so one set is derived from, that is mother, we call we get 23 pairs from like this mom and dad, like, so one set is obtained from ovum and one set is obtained from sperm. So two sets of chromosomes forms the zygote. So that's all in the process of fertilization process, and I hope you will acquire at least a bit from it. And if something in the video it is not clear or you have not understood, just you are free to ask me. Just type under the comment box that in the below, or you can send me personal message also that this one is not clear to me, or if it is not been explained properly. So don't hesitate, just ask me. And I hope you will like this video. So I just want to conclude here today with the fertilization process. I know you have already, I think you have already gone through it, but still you go through it, just revise, just you recall whether you know all these terms or not, then so the concept will be more easy to you. And so I want to conclude now. So thank you and all the best. Study well, study nicely, and okay, I will upload in the next class. I will do with another topic. Okay, thank you, bye.